Hello, it's Lindsay, and I am here with my final for microbiology. I have my questions printed and ready to go. So, question number one was, what was your favorite topic or one you feel is most applicable to your current or future career covered this semester, and explain why? My favorite topic was definitely food microbiology, and I genuinely enjoyed learning about the starter cultures and how microbes are literally used in food. But I think what I liked more than that was the decision to do my research project on food microbiology. I'm probably posting that tomorrow. Just because of all of the learning that I did on my own about the future of food microbiology, and I guess like dabbling in biotechnology, specifically how to handle widespread overpopulation and food insecurity and issues globally that are problematic. So, precision fermentation and just literally making microbes the main food source instead of like a starter culture like a lactic acid bacteria or yeast, and how you can take a microbe and turn it into a specific food source, which sounds very futuristic, but I learned that Quorn, Quorn, however you say their brand, is already basically using that, and there's a lot of money on the table for people to get the process behind that developed and figure out how to scale it and sustain it and what they can do with that. I'm vegan, and I found it fascinating that I've actually purchased before. There's a ice cream brand called Brave Robot, and they talk about being dairy free, but it's still whey protein, and so apparently they do this. And all these last couple of years since I've gone vegan of buying this, and it's never crossed my mind as to how it worked. It was just a marketing ploy to me until I really started to understand more about the microbes behind it. So that was pretty cool. Question number two is, if you had an unknown microbe, what steps would you take to determine what type of microbe it is, and are there particular characteristics you would search for? So I would start with just a good old-fashioned microscope to try and determine what it looked like, the shape behind it, if it was mobile. I would probably work on determining if it was gram-positive or gram-negative based on the staining that it took to. And then outside of the microscope setting, I would do PCR testing, the polymerase chain reaction testing to see if I could determine it that way, and then DNA sequencing to figure out the ins and outs of it and be able to match it against microbes that already exist. If it was a completely new microbe, I'm sure there would be lots and lots of new steps. But just overall determining it, I would go through the microscope first, and then work on stains and the testing after that. The third one made me laugh. Imagine you are interacting with an individual that does not think vaccines are effective. Using your knowledge of immunology, epidemiology, and microbes in general, describe how you might converse with this individual to educate them on vaccines. So I'm going to go out on a limb and say that it's probably not just a straight up anti-vaxxer, and it's more someone that's on the fence. For instance, I did my vaccine project on the chickenpox vaccine. I was born in 87. We didn't have a chickenpox vaccine. When I was growing up, we had a pox party. Myself and all my friends got chickenpox at the same time. And when my husband and I had our first child, we had talked a lot about, staying on the vaccine schedule. And then we found out that there was a chickenpox vaccine. And I asked his pediatrician, like, why on earth is this a thing? It's literally just chickenpox. And she went into telling us about, the benefits of it and the shingles correlation and all that jazz. So I'm off topic. But assuming that the person that is not thinking that the vaccines are effective. Hopefully they just don't understand and they're not an anti-vaxxer. But I would probably start with herd immunity, because I feel like, or I like to assume that people can be pretty altruistic in nature in general, like inherently altruistic. So if you just straight up don't think the vaccines are effective, for instance, like getting the flu after you've had the flu vaccine, or folks that have gotten COVID after they've had the COVID vaccine, I would bring up herd immunity and talk about how it's important to vaccinate ourselves to protect those that cannot get the vaccinations for whatever reason. If I can't get them to come to my side altruistically, I would try and dispel any myths that they were operating under the assumption of. So, for instance, you hear a lot about virus shed, or like live virus or fetal cells and viruses. And I would discuss with them if it was attenuated, or inactivated, and the difference between the two. I would also talk about what the vaccine literally does in your body. So when it comes to stimulating your B cells, and teaching it what the virus is and how to prevent the virus and identify the virus and eliminate the virus in the future with antibodies. And I wish I knew the guy's handle. There was a cool TikTok when people started doubting the effectiveness of the COVID vaccine. But he basically acted out how vaccines work in your body, and it changed people's minds about things. When it came to identifying the COVID cells, little spike-hand viruses, I'm thinking that you probably have seen it because you have a TikTok, but just really trying to determine why they don't think that they're effective, and then going from that. So you can go based on the altruism, or you can go based on just the cold hard facts. I also tend to be, I don't want to say pessimistic, but there's historically proven less deaths with vaccines, so hit them with some of the less cool facts, the harder hitting facts if they're still on the fence, and then just hoping that they're willing to listen versus shutting down completely and just wishing you away. Question 4 is, describe at least 2 positive and 2 negative relationships humans have with microbes. What are some advantages and disadvantages of each relationship? I like this question because I feel like the positive relationships with microbes have to do with like preventing disease in humans and preventing disease in crops. But then on the other side of the same coin, the negative relationship that humans have with microbes is pathogens for us and pathogens for crops. So microbes are very beneficial when it comes to disease prevention, with antimicrobials and probiotics, prebiotics, all the things that can keep you really healthy. There's obviously microbes that are also pathogenic and can make you very sick. And the same with crops. So you have the nitrogen fixing plants, and they have symbiotic relationships with microbes in their soil and in their environment. And then on the same side, you have my entire garden this summer that just brutally suffered at the hands of every freaking disease you could ever hope for, because it was too hot. And I feel like going into this course and that first discussion post with Ed Yong's Beneficial Microbes, where everybody was like, oh yeah, no, I kind of just thought that they were all disease-causing pathogens, because microbes are they get a bad rap, apparently. Clearly, I genuinely like the fact that, it's an advantage to have microbes that live in your gut, but it's also a disadvantage to have microbes that live in your gut. And it's an advantage to have microbes that are found in soil, but it's also a disadvantage to have microbes that are found in soil. So it's all about deciding and determining what is important in those instances and how to take advantage of the good and try and fend off the bad as best as possible. Question 5, the last one is, many applications of microbes exist. List and describe 5 uses of microbes. So my first use is in food application with lactic acid bacteria, yeast, all the starter cultures, and the biotechnology side of food production. The antimicrobials that I kind of discussed earlier, talking about with benefits of them in the body and disease. And the antimicrobial side of microbes is a massive benefit. Breaking down waste in not only us, but also in the water is another benefit. So the whole module on, or the section of the lecture on water purification and breaking down all of the negative oils and things that needed to be cleansed and purified out of water. I think of an old school commercial where you see these little, they just look like bugs in the commercial, but they're microbes breaking down things at a septic tank, and keeping all of the soil and water cleaner than it would be otherwise. Insecticides is another application of microbes, and I've read about BT, which I can't remember the name of the longer form of it right now, but the abbreviation BT, and how it's basically an organic pesticide, and eliminating the threat in agriculture when it comes to microbes, or using microbes to eliminate the threat. And then our microbiome is another fascinating application of microbes. And the gut microbiota, I've always kind of been cautious or iffy about supplements and things that people tell you that you should take that I'm just pretty convinced you can find in food. For instance, like calcium, vitamin E, etc. However, the more I read about all of this, and I haven't had to take antibiotics in a really long time, but I know the next time one of my kids has to take antibiotics, they will definitely also be taking probiotics immediately. My oldest had a double ear infection recently, and he had to do 2 rounds of antibiotics. And I'm so sad that it was when I was in the first 8-week chunk of time instead of this one, because I feel like I would have treated that entire thing differently. But yeah, lots and lots and lots of applications of microbes. Ed Young's book was a fun addition to this course, and I genuinely enjoyed being able to read that and see somebody else's perspective on it, and it led me down a rabbit hole of a lot of TED Talks. So that's pretty much it. I feel like that was all pretty high level. I feel like I could talk forever and ever. I know we have a 15-minute limit, but I really enjoyed the course, and I really enjoyed the subject matter, and I don't think I realized how much microbiology and biotechnology and everything that it entails would pique my interest this term. So thank you very much.